Hello everyone, my name is Jyoti Sharma. Today I'm going to present my work on discovering the suppressed resistance in wheat lines for stem and leaf rust diseases. Before going into the specific project, let's discuss what is the suppressed resistance. So in the wheat, there is three subgenomes for A, B, and D, and it has been reported that sometime when the gene has been transferred from the diploid resource and from the tetraploid resource into the hexaploid wheat, there is loss or there is a reduction in the functioning of the resistance gene. So that is known as the suppressed resistance in the hexaploid wheat. So whenever the D genome is removed or the suppression associated with the D genome removed, the line becomes the resistance. So there is example for that one, the hexaploid wheat cultivar known as Canthach, which reported to be carry the suppressor gene SUSRT1 on the 7DL, and it is susceptible for stem rust. But whenever the suppressor gene is mutated and the D genome has been removed, the Canthach tetra it become resistant. So that explains the suppressed resistance. So now we are moving towards the specific project in my current study. The current study was done to identify the suppressed resistance gene against two important fungal diseases in wheat, stem rust and leaf rust diseases. These are caused by the Piccinia species. So the stem rust studies, we use the Canthach mutant line NS1, which help to understand which gene has been suppressed in the Canthach line. And for the leaf rust, we want to identify the Stewart 63 genes, which lost when it has been used for the development of the synthetic hexaploid wheat line. So this study was conducted on the double haploid population developed by crossing the resistant line with the susceptible lines, and then which help us to identify for the stem rust there is 4 genes present in the Canthach mutant line that's present on the B and A genome, and similarly for the leaf rust study we are able to identify the resistance gene location on the 3AL and 5S chromosome. So we are conducting further analysis to understand more about these genes. So based on our ongoing studies, we came to this conclusion that it is important to understand what happened when the suppressed gene removed or they mutated. For example, in this case, when suppressor gene has been removed, it's untangled different expression pathway and our genes expressed, so that line become resistant. So it will definitely gonna help for the future wheat improvement. With that, I would like to thank our collaborators and our funding resources. Thank you.